Bioinformatics.
Okay, so welcome to the last part of our RNAseq lectures. And in this one, I'm going to kind of build on where we stopped last time. And so to refresh your memory, we ran through the basic differential expression analysis. And that left us with a big table that could be 30 or 40,000 rows of different gene IDs. Most importantly, the log fold change, in other words, how big of a difference was there in the expression level between our different treatment groups, whether it was drought versus well-watered or whatever kind of treatment you're looking at, and the directionality of that is indicated whether it's positive or negative. And then the adjusted p-value, again, a p-value that accounts for a false discovery rate correction. And so what I want to talk about today is two things. One, how do we go from this table and start putting together something that has some biological relevance beyond just a list of gene IDs? And then what are some caveats for these types of experiments that should be considered in the future? So one of the things that we're typically going to see happen with these types of analyses is one of the very first kind of post-differential expression comparisons people will make is to take this list and do what's called an enrichment analysis. And so to talk a little bit about an enrichment analysis, I need to talk about what is required. And so you've learned at this point how to make a genome assembly and how to annotate a bacterial genome. And so in order to do an enrichment analysis, you need annotations. In other words, tags on all of your genes indicating what they are or what they are for, whether it's involvement in the Golgi apparatus or photosynthesis, or even as specific as being a component of making photosystem 2. These things are associated with different gene IDs, and they are associated with different functional categories at different levels. A very popular way of annotating functional function is through gene ontology annotations. And so gene ontology is a hierarchical classification of typical gene molecular functions. And so to kind of illustrate what I mean by hierarchical classification, let's look at this example of pants. And so if you had a number of different categories you wanted to think about pants in, there could be different levels to it. You could say, hey, these things are just pants. Any pair of pants could be classified as pants. But you could have pants that are classified at the store as men's pants, if you want to go down a level of specificity, women's pants, kids' pants. Looking at men's pants, you could have casual pants. A subtype of casual pants could be khakis. Those aren't casual for me, but that seems very dressy. But other people might classify dress pants as including only trousers. You could also have sports pants. Again, I don't know what those are. But you kind of see the point how if we were to look at a pair of trousers, they could belong to both the trouser classification, dress pant classification, men's pants, as well as pants. So you can belong to multiple categories within a hierarchy. And functional categories for gene ontology are the same way. We've got major groups like one major group is biological processes. And within that, you could have cellular physiological processes. Below that, things involved in cell death or apoptosis. You might have things involved in intracellular signaling, which is a form of cell communication, which is a cellular process. And so you can have these types of annotations for all of your genes, all the way down to as specific as we're able to get. If you're working in a non-model system, chances are you don't have annotations yet for your species. Bacteria are relatively easy to annotate. Eukaryotes a bit more challenging. That said, you can actually perform a transcriptome assembly using all of the RNA that you sequence for your differential expression comparison, and annotate those individual transcripts you assemble using something like trinitate. This is way beyond the scope of this class. It can take weeks to run in some instances, but it's a fairly straightforward pipeline that yields GO annotations or gene ontology annotations in novel systems, so you can do enrichment analyses. Okay, so let's talk about how we actually run an enrichment analysis. So if I have a list of genes that I've pulled out from my differential expression comparison that were significantly different between our treatment groups, as well as their assigned gene ontologies, You could have multiple entries for the same gene because you could have multiple gene ontologies for the same gene. Again, hierarchical classification. And so what an enrichment analysis is going to ask is, in this list of differentially expressed genes, are there functional groups or gene ontologies showing up more frequently than we would expect by chance had we pulled that same number of genes for that list at random from our original species-wide set? And this is a enrichment analysis. And so to kind of illustrate that, let's imagine there's an example where our reference, in other words, the whole genome or whole transcriptome here in green, and then we've got our differentially expressed genes here in this kind of dark blue or dark green color. There are 5,000 total genes annotated in the reference, and about half of those, 2,500, were called as differentially expressed. In other words, for any functional category, we might expect by chance that if there were 200 genes involved in homeostasis in the genome, and if homeostasis was not something enriched for in our analysis, that just by chance, if we were to pull 2,500 genes out and as a list, we would expect about 100 of them to be involved in homeostasis, meaning that it didn't seem to vary in its frequency whether it was involved or not involved in that differential expression comparison. And so here's an example where we can see the total within the genome and then the total within that half of the genome list that was significantly differentially expressed between our treatment groups. And so how do we go about knowing whether there is a significant enrichment or not? And a simple way to check for this is a Fischers exact test. So this will tell us if the number of differentially expressed genes significantly departs from our null expectation of carrying over the frequency based on the size of the list. So now we've got a new column here that's the expected number of genes involved in a given GO category if we had just pulled that 2,500 gene list at random. And so here, homeostasis, we expect, again, about 100 genes. We actually only saw about 52. MAP kinase, we expected about 25. We saw close to 50, almost the full amount of MAP kinase genes. And so we can now statistically test whether this actual observation significantly differs from the null expectation. So this is math you'll probably never have to do by hand, but if you wanted to, you could do it. You could enact a Fischers test within R, for example. In here, we're building a table that looks like this with MAP kinase, non-MAP kinase genes, and then total genes. So differentially expressed, there were 45 out of 2,500. And for MAP kinase that were not differentially expressed, there were 10 out of the remaining 2,500. So we could just do a quick Fischers test, make that matrix right here simply, and test to see if it is greater than we would expect by chance within this cell. And if we ran this test, again, you're not going to do this by hand ever, but we get a very small p-value, and we would call MAP kinase as being significantly enriched for being differentially expressed in this data set, meaning that MAP kinase functions are probably somewhat related to whatever the response to this treatment or grouping that we compared in the first place. So because we're not going to be doing things by hand, there are many, many different ways to enact these gene ontology enrichment analyses. GoSeq is one that you can enact in R. There's also web-based ones like GoRilla or AgriGo if you're interested in agricultural genomes. So these are all kind of fun. They all have names with the word Go in them for gene ontology. So here's a actual result from a study that I did. And here you can see a bunch of gene ontology IDs, as well as the p-value for overrepresentation, meaning it was higher than expected, and the p-value for if it was underrepresented, because you can have things over-enriched or under-enriched. So in this case, this one was significantly overrepresented, and kind of the opposite of that is there's definitely no chance of it being underrepresented if we're very sure that it's overrepresented. We had about 167 genes involved in functions related to chloroplast thylakoids out of a total of 367 in the genome. And if I recall, there were only about 20% of the genes called as differentially expressed. So this is a significant over-enrichment. Now, what can we do with this type of information? It depends what you as a biologist think is interesting. In the instance of this work, we noticed that almost all of the functionally enriched categories had to do with plastids, the chloroplast, for example, and photosynthesis that were enriched. And this was very interesting to us because it had to do with plants that had fewer cells but were maintaining equivalent rates of photosynthesis across a unit area of leaf tissue. So it made a lot of sense biologically to us. If you're not interested in gene ontology, that hierarchical classification, you can also, in better model systems, look at pathway analyses. These are very similar. KEGG is a popular example of this, where you can look at genes that are all involved not in the same function, but are involved in the same pathway. So they can have different functions, but are connected in some intrinsic way, like photosynthesis. You've got photosystem 2, photosystem 1, all this fun stuff. All these different genes are involved along that process. And so you could look and see if gene involvement in certain pathways or processes are over or under enriched as well. You can even go a step further, and here we've got a kind of abbreviated cell cycle kind of pathway, and you can take your log fold change, and in green say it's higher in your treatment A, and in red say it's lower in your treatment A, and you could actually visualize not only genes that are significantly different across your pathway, but the directionality of the difference. You might expect that sometimes there can be compensation. So here we see no change in this gene, we see upregulation in this gene and downregulation in this gene. You might expect that that could kind of wash out differences downstream. But here we've got kind of consistent downregulation of whatever is contributing to this process here, leading to upregulation here. So there's lots of different ways you can kind of look at these things and get more biological inference out of your data set. So the last thing I want to talk about is something that's not talked a lot about, but it's some important caveats related to RNA-seq data. And it's something that, as a community, there has not been a great deal of effort to correct for. So when we do our differential expression analysis, oftentimes you'll hear things like, oh, this gene was expressed more than this gene. But that's actually not the case. It's important to understand what your analyses are actually doing. And so if we look at what gene expression is being compared as, again, we're starting with mRNA in our samples, we're extracting it, we're fragmenting it and sequencing it through Illumina, whatever you want, and then we're taking those reads like you have done in this class, and you map them to a reference genome. We then use read counts, in other words, count the number of reads, right, that are mapping to a given region, and using that to then go and do our downstream read normalization, right? Reads per million, or fragments per million, or transcripts per million, whatever you want to do. And so something that's important to keep in mind is that we're not actually counting the number of transcripts from that species. We are counting effectively, because we're dividing by the number of millions of reads that we sequenced, the proportion of reads for a given region relative to the whole. You can think of this as analogous to concentration or even molarity. So when we are comparing gene expression differences, we're saying per transcriptome, or per sum of RNA that was sequenced, there is a difference in the proportion of our gene of interest relative to that whole between our two groups. And bear with me, there's a reason why this is an important distinction. Okay, so as an example, let's imagine a perfect world where I know exactly how many transcripts are being produced by things before I even sequence them. And so in this example, we've got sample 1, both what it's actually producing and what we are going to be inferring from our RNA-seq comparison, and sample 2 for the same thing, both the absolute abundance that we know the cell is producing versus what we're assuming when we do transcripts per million. So this is a simple example. There are 6 genes. In sample 1, gene 1 through 5 all express 20 transcripts, and gene 6 has 100 transcripts. In sample 2, Same thing, except the final gene has 300 transcripts. And so what this means, if you look at this, if you were to take the 20 here and divide it by the total number of remaining transcripts, it makes up about 10% of the transcriptome. In other words, its concentration is 10%. The 20 here is the same number of transcripts being produced by the cell, but because there is another gene producing so many more that it's inflating the size of the total RNA pool, we're only going to assume that it's half the concentration. So if we were to actually compare the expression level of gene 1 between sample 1 and sample 2 in a differential expression comparison without doing anything special, we would think that sample 1 is expressing gene 1 twice as highly as sample 2, and that is simply not the case. It is at a double the concentration, that is true, but it is not twice as much transcription happening. So this is a very important clarification to make because this can impact the biology of organisms. Another way to look at this was really highlighted by Lovin et al., where if you were to, again, have a bunch of genes looked at here, there's 10 for this orange species and this black species, and then if you do this kind of transcripts per million normalization type thing like we normally do, where you divide by the library size, you would take all these absolute differences in expression level for all these genes, and you effectively collapse them down. You bring them down because now they're back to concentration, and if you actually calculate the log-fold change between these, you'll actually see that despite starting lower across the board, some of the genes, gene D, E, and F, are actually called as being increased in expression relative to the orange. And that, again, is simply not the case. So why do I bring this up? Some of you may be aware that I study polyploidy or genome duplication, where you go from 2 of every chromosome to 4 or 8 or 12 or 16 or whatever. And something that's always interested me is when you add extra chromosomes, do you add extra RNA to the total pool? In other words, do you increase the pool such that concentration is not a thing that you can assume to be similar or comparable between your different treatment groups if you're comparing a diploid and a polyploid, for example? And this has, not been looked at very much. There's some examples in soybean, for example, but it's not just a polyploid issue. If you're comparing different species, if you're comparing maybe chimpanzees and bonobos, I think those are different things, they might have all the same genes or very similar genes, but are those genes all producing across the board a similar sized pool of RNA? Because if they are not, then you're not comparing absolute expression level, you are comparing concentration of expression level. This is also important if you're comparing large stressors. Again, I go with plant examples right now, but if you were to induce drought, this can prompt abscisic acid to be produced, and this is a critical signaling hormone that can actually facilitate change in over half of the genome, in wheat at least. And so if you're increasing expression around half the genome, then that could also be inflating the pool. So it could be the same species, the same ploidal level, but if your stressor has wide sweeping effects, it could inflate or deflate the transcriptional pool as well and make some of your comparisons, again, purely concentration based. Luckily, there are ways to correct for this. They aren't used very much, unfortunately, but it's as simple as adding standards. It's funny that in so much of biology we use standards, but when we get to next-gen sequencing related things, all of a sudden standards go out the window in many cases. And so Lovin et al. Actually proposed this really kind of clever mechanism of using synthetic RNAs to add known concentrations of RNAs to your samples right before you sequence them, and adding them equivalently. So here they are as the triangles. So these orange and black triangles were added in the same concentration before sequencing. And so, yeah, your samples get sequenced slightly differently. And so the triangles reflect that. You would expect these triangles to be right on top of each other if everything was sequenced equally. That's why we do normalization in the first place. So now, rather than normalizing by the whole data set, you can divide by the library size of just the triangles to pull things down just enough to account for differences in the sequencing bias, but not for the actual transcriptional kind of total size differences that are biologically there. And now you can pull out actual log fold changes that reflect the original differences. And this is something very important to consider. However, there's a reason why people don't use these methods as much. One, they're expensive, they're an extra step. The other is that they're not robust to changes in cell number. So what do I mean by that? If you have a treatment that influences cell size, there's lots of things that do this. Polyploidy is, again, an example. You might think of a leaf where polyploidy has larger but fewer cells. So now you have to think about another angle of how your comparisons of transcription can be influenced by how you normalize. Because if polyploids have larger but fewer cells, and you want to compare transcription per cell, you have to now account for something else. So let me give you an example of how different ways of normalization can tell you different things when you have kind of variation in cell size or cell number. So again, let's say we're interested in this gene that produces this red transcript. We've got polyploids that have larger but fewer cells that also produce more total RNA relative to their diploids. So we could spike our total RNA that we extract from both of these samples with the same number of these triangles or spike in standards. And this will enable us to do a lot of different normalizations. There's no reason to do just one. We could ignore the spike ins, remove them from our data set, map them out, and do a regular differential expression comparison where we take the volume of our transcript of interest and divide it by the library size, basically. So this is a transcripts per million, kind of a normal library size normalization, and compare those numbers. In other words, we would be comparing, again, the concentration of red relative to between these two groups. And I call this, and others call this, a per transcriptome comparison, because we're comparing whether the difference of this red relative to the rest of the transcriptome, so red per transcriptome, is changing. And here, it's not changing because there are two for this pool, and this pool is twice as large, and there's twice as many for this pool. So there is no significant difference per transcriptome in this example. Here, we're not going to account for cell differences. We just extracted from equivalent volumes of tissue, spiked it based on that volume of tissue, and then normalized by dividing the total transcripts sequenced by the total of the spikes that were sequenced. So we're basically, again, just pulling together by those triangles. And now we are basically comparing the number of transcripts per unit of biomass between these two groups. And here we're going to see that there were 6 across these 3 cells within this unit of biomass, but there were 8 across these 2 cells within this unit of biomass. So we would call this as being significantly higher expression per unit of biomass. Finally, we can actually factor in differences if we know the cell density differences between our samples. There's lots of ways to measure this. And if you have that info, you can do the exact same thing as the per biomass, but apply a scaling factor to your spike in abundance prior to dividing your library by it effectively. And so here we could multiply this by 3, the abundance of these. We could multiply this by 2 and make that number reflective of the difference in cell contribution, so that now we are basically comparing the number of transcripts per cell, or an analog for that. And here we would see, again, this one would be higher because there are 4 per cell versus 2 per cell. And so with just kind of adding one basic methodology and thinking kind of critically about how to use the output of that methodology, you can really start to make multiple comparisons that are all individually biologically informative and also more specific, rather than just saying our genes were different. And so what this kind of looks like as a workflow, if you're thinking about how kind of big picture these things work, you know how to do a lot of these things. you guys know how to take raw reads and do quality control and trimming and taking off adapters. You know how to take your cleaned reads and map them. I didn't show you how to actually pull out the read counts from the mapped reads, but that's actually relatively straightforward. You can take a reference from your spike ends and map to those as well. So you have both the mapped reads to your genome, the mapped reads to your spike end, and then you can use those two pieces of information to either normalize per transcriptome, per biomass, or per cell, if you have cell density data. And then you can do all of your downstream analyses from the output of that, your gene functional enrichment analyses, all sorts of fun stuff. And so in this case, we actually found a big difference when I did this with my last diploid polyploid system. We found that we were missing a lot of information when we were only comparing concentration-based differences. Per transcriptome, we only saw about 2,600 genes being differentially expressed. Per cell, we saw about twice that. And what was a kind of a cool way to compare these pieces of information was to think about genes that are being upregulated per cell in the polyploid, because they have fewer cells per leaf area, just enough to be not differentially expressed per biomass. So there are certain genes that we identified that were upregulated per cell, just enough for there to not be a difference when you're looking at kind of the size of the leaf across that unit of biomass. And when we looked at those genes and performed those kind of GO functional enrichments that I showed you earlier, the genes that were actually doing that were being upregulated to maintain consistency across the unit of biomass, were genes that were over-enriched for functions related to photosynthesis. And that made a lot of sense to us because per unit leaf area doing physiological work, there was no difference in photosynthetic output between these plants, despite these cell differences. And so it was kind of interesting to see that through this combination of methods, we were able to validate that per cell, there seemed to be higher upticks in photosynthetic throughput through the upregulation across the board per cell of photosynthesis-related genes and their functions. So that last bit is there for those of you that are going to be going off and doing RNA-seq in the future. I think, again, these normalizations and the caveats and the biases are things that people really need to consider. This is kind of my soapbox that I feel very strongly about, and it's largely gone ignored by big chunks of the community, and so I try to educate people in this class to at least think critically about it. And when you read a paper in RNA-seq results and they say the genes were more highly expressed, you need to stop and think and go, no, we're scientists, we need to be a little bit more specific. They might be in a higher concentration, but we don't have the information to say that they're more highly expressed. So if that was a bridge too far for you and you're not planning on using RNA-seq in the future, hopefully you can at least read papers that use RNA-seq, because again, it's one of the most popular methods these days for a molecular perspective. And ask yourself, did they properly replicate it? Was it a controlled environment? Were they able to actually make quantitative comparisons, or were they qualitative? Did they make quantitative comparisons without having the minimum number of reps and doing it properly? And how did they put those results into meaningful contexts? And so hopefully this will give you a little bit of usable information to go out and read those papers, because there are a ton of them that use these methods all the time.